My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. We are continuing our student perspectives series, and today we have a very interesting guest in that we interviewed his advisor a number of years ago, and his advisor is Dr. Stephen Mackessy, who does a lot of Venom research. I would encourage everybody to go back and listen to that. But we have Sam Kerwin today, who is a PhD student in the Department of Biological Sciences at the University of Northern Colorado. He's also a curator of reptiles for the museum there. And so we'll talk about that as well. But as everybody knows, I love venomous snakes, and I love venom and all topics around that. So I'm really excited to talk about that today, but from the perspective of a current student. So welcome to the podcast, Sam. How are you doing today?
I'm doing very good. Thank you so much for having me on. It's a great opportunity.
Yeah, it's great to have you here. So as you may know, I know you've heard some episodes. I like to get the story from the very beginning. So I want to know, snakes in general are kind of this eccentric thing to a lot of people. And even, somebody who's really into venom and venomous snakes is probably a lot more outside the box for people. So how did you originally get into snakes? Is it something from your childhood or something later in life?
It definitely started from the childhood. I grew up in rural Illinois, where we're not known for our abundant venomous animals, but I grew up outside, like probably many people that have been on the show, and probably yourself as well. Just having the ability to have your hands dirty as a kid and not be scolded every time you came in the house with blood on your feet. I think as a kid playing in the dirt, you get these dopamine rushes from finding the rarer animals, or the animals that are the ones that deserve your respect a little bit, I guess. So, I mean, the ones that were noticeable for me back home were, if I found a big copping snapping turtle in a big pond, and finding that animal is so cool because not only is it something you don't see very often, but also it demands your respect. And it, definitely an animal that warrants some distance and a little bit of, just caution. And I had a lot of family living in Arizona, and so I would spend summers there as a young kid. And, if there's any animal that kind of fits the definition of deserving your respect, it's something like a rattlesnake that has really leveled the playing field from, a biochemical perspective with humans. And I found that so fascinating. And I think that kind of interest in those types of animals, and then maybe some family members not nipping that interest in the bud soon enough, kind of just spiraled. And it just led me down a pathway of, where can I possibly do more of this and, and see more of this stuff. So definitely from a childhood age was super interested, and, even from a childhood age was super interested in the venom aspect too, which has taken me further than I ever thought it probably would.
Yeah, well, it's great that, we, relative to people's families, we hear, it's kind of all over the place, but we do hear a lot of, family members who were kind of anti a childhood interest in snakes. So I always like to hear when somebody's, family, didn't kind of put an end to that childhood interest. So I'm glad that was what happened in your case. Yeah, I was going to ask you. You mentioned Illinois. One's North American snake. I don't think it's particularly rare, but I've never seen that. I've always wanted to. I just need to go do it. But is a fox snake? Did you have fox snakes where you grew up? Did you see them often? Or?
I grew up in really, really dense agriculture. And so, finding snakes was super rare. I mean, every once in a while you find like a, Decay's brown snake or just a garter, and that was about it. We never really saw much more than that up there. I mean, granted, I was too young to ever like really put some serious attention into finding them, but now that I'm older, I want to go back and find some of the stuff that I probably missed as a kid.
Yeah. Yeah, that's great. So you have this childhood interest, and you grew up in Illinois, being inspired by things like snapping turtles, and even kind of taking an interest in Venom, as you mentioned. When did you realize, when you were a child, that this is something you could potentially study in college, something you could do as a career? And then, where did you do your undergrad? How did you get in there? Again, did you start in biology? Were you going to be an insurance salesman and had some epiphany moment? How did that all come about?
I was pretty one-tracked, I think, my entire childhood. Like, I knew fairly early on that I was going to do biology in some way. My family in Arizona, despite their reluctance to like actually participate in any of this interest, they, very quickly found that there was a facility in northern Scottsdale, which is the Phoenix Herpetological Sanctuary. And, when I was about 14 years old and I was visiting them for, a holiday, they took me up there, and I realized, I was like, oh my God, I could volunteer here as a kid. And I had to like get them, on board with the idea, but they ended up like driving me back and forth every day. And I was able to just kind of be around more animals and kind of see the field a little bit more seriously. And that really took off. And I ended up spending, almost about 7 years at that facility, kind of working my way up, working with a bunch of animals. I ended up working with the venomous collection about 17 years old. And the whole time I was there, I knew eventually I wanted to work with the venomous snakes. Like that was the goal the whole time. And I think it all just kind of stemmed from this idea of like these animals are doing something so fascinating. And I want to kind of be more familiar with them, with the goals of understanding what's going on. And so that then translated to my undergraduate work at the University of, not the University of Arizona, but Arizona State University.
They just played in football too. You just almost made a big mistake there. Anyways, go ahead.
I'm sure some my old professors might be cursing my name right now. But I did Arizona State University. I did my undergraduate there in applied biological sciences. And I would spend my weekends working at the sanctuary and, having the ability to work very closely with animals I probably should not have at that age. But it turned out well. And then once I graduated, if you look up venom research laboratories in the United States, there are not many that come up as frequently in your search bar as Dr. Mackessy's lab.
Yeah, certainly.
And, I sent him an email, and, he happened to have a position open for a master's student and jumped in. And I just kind of, went right into the deep end.
So going back to the undergrad, though, before we talk about the, the graduate work. So you're at which Arizona school now? You're at Arizona State?
Arizona State. Okay. Arizona State.
So you're at
Sun Devils.
Yeah. So you're go Sun Devils, right? No. Anyways, so you are at Arizona State, and you're getting, you're just diving in and really just taking in everything about snakes. You're working at the sanctuary. I'm sure, studying these animals. I'm assuming you're going in the field in one of the best places in North America for rattlesnakes in particular. And so within like if you just start with snakes, meaning like I'm interested in snakes, I'm going to study biology. There's so many different directions you could go. You could be studying community ecology or behavior, or you could be a conservation biologist, or you could be this or that or the other thing. And you already mentioned the venom interest when you were young, but was there something during your undergrad that really like propelled an interest, not just in the venomous snake? Like, I worked on venomous snakes my whole life, but I've never done a single project related specifically to the venom. So how did you get to the venom part of the venomous snakes? Did that happen during your
You know,
undergrad?
it's interesting because I'm pretty sure, like as far back as I can remember, the interest in Venom almost came first. I think like there was definitely a part of it that almost superseded anything else. And I remember, just being able to work at the animal sanctuary, and every once in a while, we would get called to help with, moving a crocodile from one pen to another, or, working with some large monitors or doing stuff with tortoises. And, I loved working with animals, period. But there was always just something almost mysterious about the venom. Like unless you take the time to really really study it's kind of this amorphous thing to you, and yet it still has such a huge kind of presence in how we talk about reptiles, and especially how we talk about snakes. I think when most people think about snakes, the venomous ones are at the forefront of their mind, and so it's despite not being shared among all reptiles, it takes such a huge part of people's kind of thoughts when they think about these things. And, like I kind of mentioned, unless you really study it, it's just amorphous concept. And so I think that interest was there from a really, really early age. And I honestly, I saw undergraduate work as a pathway to maybe understand what's going on a little bit better with venom, like, which is kind of a strange way to think about your undergraduate degree. But I was like, maybe I'll take biochemistry and that will help me understand what's going on. That's got to be the key to this, right? Or I'll take, molecular genetics, or I'll take, organic chemistry, and just so I could maybe get an inkling of what is happening within this weird substance that these animals use.
Interesting. So you were right through the undergrad. You were focused almost more so on the venom, and even taking your coursework to prepare you to learn more about that specific component. So that's interesting. So you mentioned looking for venom related labs, and maybe your story is kind of a short one, but with these student perspectives, I do like now and then to kind of get some advice from students on different stages in their career. And I like to ask about the idea of how you found your graduate program, how you got in, because this can be very difficult to get into a graduate program. And so and a lot of people don't have much of an idea in terms of how to find what they're looking for. Again, it sounds like your story's pretty focused and narrow, but I'd just be curious that process, how you found where you wanted to go to school, what the process was like for reaching out and getting getting the go ahead to go to school there.
Yeah, I found that probably the best way that I have found interesting labs, or labs that at least have shared something that I may be interested in, is looking at like the works cited of literature that you find of interest. So not necessarily looking for typing in Google, but like a venom lab, but typing into Google Scholar, like, hey, I want to look at papers that cover these topics, and then looking and saying, okay, who's doing this research and where are they? And that tends to be a bit of an easier way to kind of sort through the mess that is, any search engine result nowadays, where you get, the first half a page is all sponsored stuff. And then, the second half of the page is all like these kind of eye-catching news stories of, events that may or may not be related to what you're actually trying to do. And so I kind of went in that route, and, kind of, again, it's hard to not see Steve's name pop up on almost every like interesting venom paper. And I was also very fortunate. I went to the Biology of Snakes conference at the Chiricahua Desert Museum in 2017 when I was, I think I was about 16 at the time. And I was, I was lucky enough that where the sanctuary sent me, and, they were more than happy to fund it, which, as a 16-year-old interested in snakes and venom, I'm like, oh, this is amazing. And I got to see two individuals from Steve's lab actually give presentations. And, it was a snake conference. And so there was a lot of ecology talks and population dynamics and everything that we normally, associate with these guys from a behavior perspective to ecology. And then you had a couple talks that were on like venom. And one of them was Dr. Kara, well, now Dr. Kara Smith. At the time, she was still in Steve's lab as a student. And then now Dr. Melissa Bernard Valle giving talks from his lab. And they were both, fascinating, absolutely fascinating talks. And that really kind of solidified, my interest in his lab, just like the idea that this awesome work was coming out of his program and these were students was so cool to me.
Mm-hmm. Yeah. So a couple interesting pieces of advice there. Don't necessarily just Google search, labs of various faculty members. Mm-hmm. Actually go to the products or the scientific papers, and there's many ways to do that, but probably the most accessible way to the most people would be going to Google Scholar, and you can really just narrow down who's doing what pretty quickly.
Yeah.
I like that. And then also just the concept, if you can, going to a gathering of some sort where a lot of these people will be, and really, kind of fine tuning your interests and thinking about who and communicating with who you might end up wanting to go to school with. So all good advice. So you end up going to Northern Colorado, and you're coming in as a master's student. And how, so I've talked about in previous student episodes, there's different situations that people come into. You might come into a graduate program, for example, where a faculty member has a long-term project, and you have kind of a little bit broad guardrails in terms of what you want to do, but it has to be within that system, within the long-term setup. There are other people who you come in and it's a funded project, say by a government agency, and X, Y, and Z need to be done, and the money's there to do it. And there's, faculty members that bring you in, and as long as it's within a very broad topic like Venom, say, you develop your own project, and there's all kinds of variations in between. So I'm just curious, when you decided to go and were accepted into the university there, what was the situation for you? Did you just know I like Venom, or did you come into something very specific?
I definitely came in with a very general idea that was very quickly shot down because I realized after, I think, one short conversation with Steve, we were like, this would be a 5-year project, and I am not a 5-year student at the time. So if the kind of initial thoughts changed rather rapidly, and Steve's lab does certainly have projects that are kind of tailored towards, master's students, and they're only designed to take a couple of years. For example, if you pick, let's say, one species of venomous animal X, Y, or Z, and you look at venom variation across its range, right? You may know the general venom phenotype that we see in that population or that species as a whole, but let's really sample thoroughly and look for, variations. And can we find stuff, period? And can we find stuff that may influence, how we handle them from a medical perspective as well? And so that kind of protocol and that method is fairly set in stone on how we would do a study like that. And so often lends itself to an easier project. Something about Steve and I just chatting made us decide that we would go the entire opposite direction and try to put together this project that had almost zero precedence and test drugs that had never been used on animals before with technology that had never been used on animals before. And it was kind of a pain, a troubleshoot along the way. Just every month seemed like another issue had arisen. But so I think originally coming in with a general idea, but at least some idea. And then very quickly it devolved into, do I want to do this type of geographic kind of work? Not really. And then it went kind of full downhill into, I say downhill, it ended up being cool, but fully downhill into this kind of new territory that neither Steve nor I had kind of gone into. So there was an evolution of kind of the situation as we went along. But definitely started broad and then ended up going narrow for the kind of my first couple months of just talking with Steve and just having these lengthy conversations.
So how would you start with that broad? How would you summarize for your master's what you were thinking and what kind of the plan was when you were thinking broadly.
So I wanted to study venom ontogeny, and this is a topic that I know Steve kind of touched on his episode a while back, where we do have venoms that change phenotypically from young to old snakes. We see different patterns of that variation. We see different toxin families kind of rising and falling. We may see the change happening at different time periods of the life, but we don't really understand like the exact mechanisms behind why. And so my idea was just trying to investigate that. And we realized, we would have to raise snakes from babies to adult. And that was like the big limiting factor time wise. And, master's students don't have the full time commitment to really sit there and, raise them up from babies all the way up to adulthood.
Hey everybody, I just wanted to take a quick break and tell you that the Orianne Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals, but these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.orianne.org to learn more about our work and how you can get involved.
In a very general sense, again, we have a very general audience here. We understand that in multiple species, snake venom can change across the life, the age of a snake. Like, what's the mechanism there? Do we know if it's like, okay, when this snake species hits age 3, its venom changes, or is it something about like, as their diet shifts, it starts to trigger something chemically? What's the mechanism that triggers those shifts? Is it the diet? Is it a hardwired thing? Do we know?
That's kind of the thing I was looking to investigate. There's probably some good correlation to hormone levels within the individual animals. Like once they're, coming up on sexual maturity in some way, they may start shifting the actual gene expression of the, different toxin families. There are certainly very, very good examples of diet correlating really, really well with ontogeny, but it's not with everybody. I mean, just like almost every aspect of biology, there's no one rule. You always find exceptions. Like there are certainly snakes that, we don't really know much about their diet, but we see like a really strong ontogenetic shift in the venom. And so then that begs the question, is the diet the thing, or is there some other factor? Is it just size? Is there hormone levels involved? So long story short, we're not really sure. It could be one of a million things, but definitely seems to track with snout to vent length. So the size of the animal definitely seems to be a pretty good precursor for ontogeny.
Yeah. Huh. Okay. Interesting. So that was kind of the big picture that you wanted to do. That was going to be the 5-year plus project, and then you had to narrow and focus in. So tell me a little bit about that process and where you guys ended up landing in terms of your master's thesis.
Yeah, we kind of tossed that to the side, but we tossed it aside pretty quickly, but I clung on to the idea of venom production. I really wanted to know more about how venom is produced, what are the mechanisms behind it, things like that. And I got really interested in that pretty early on. And Steve kind of tossed my way a paper that he had recently published in 2020 with Blair Perry, Drew Shield, and the rest of the Todd Casto genetics lab, where they looked at the genes that were being transcribed, or like which actual genes are being kind of expressed at a given time point. And the time point that they were looking at was the actual venom production period. And there's, obviously when they're producing venom, they're, expressing venom genes. They got to make it, they got to read the genes off, they got to produce the proteins. But there's a whole suite of other genes that are being expressed that are not even closely related to toxins. And it kind of left a big mystery in the air of, what is actually going on here. And so Steve tossed that paper my way. we looked at it a lot more, and we just kind of started pitching questions back and forth. And we ended up on, kind of focusing on a question of, how do they produce venom safely? And I think that kind of took the direction, or kind of took the steering wheel and like led us down that path for sure.
Mm-hmm. And what do you mean? Well, so first of all, so I'm just kind of throwing out being cute here, but like, so snakes aren't always producing venom. And is it a start and stop type of process? When a snake's venom glands are full, can they just store it indefinitely? Does it somehow degrade? How do they get rid of it if they're not going to inject it? Just talk a little bit more about that, like the production of venom, like how it works. Like, they're not producing it all the time is kind of the question I start with.
Yeah. So what we understand at the moment is that the release of venom, whether it is like us manually extracting it, whether it's the animal striking prey or defending themselves, the release is a parasympathetic or kind of an inverse kind of trigger to the production of venom. So when they strike something, when they excrete venom in some way, they begin the upregulation of all these different genes that kind of initiate the production of venom. And so over the course of about 48 hours post, biting or something like that, we see the upregulation of like 2,500 different genes, like really, really crazy processes going on in this tiny little gland that's right next to their head. And it really kind of underlines the amount of cellular work that has to happen for them to produce these toxins. And so that usually lasts about a week or up to 2-ish weeks, depending on how much venom is actually excreted. And we see kind of this slow, gradual decline, both genetically as well as morphologically. You can actually see the cells themselves shrink back down to a smaller size once they're done producing everything. And then there's kind of this steady state kind of venom maintenance period. And that can last almost indefinitely. I mean, I'm sure all of us have, seen snakes that don't eat for months and months and months at a time. They're keeping that venom locked away, but it's ready to use at a moment's notice, which I think really, really sparked some of our interest in this work.
Interesting. Well, continue on with the work. So you're focused on venom production. And so what did you develop as your project?
So an observation that Steve had made about a decade prior was that venom out of, let's say, a rattlesnake, pick a rattlesnake, whatever. In this case, let's stick with our model species for the study, which was Proteus atrox, the western diamondback. The pH of that venom is roughly around 5.5 to 5.6. So on the acidic side, nothing too crazy, but fairly acidic. Steve had a collaborator in Africa with a puff adder who, out of curiosity did the same thing and found that the pH of the puff adder venom was around 5.5 to 5.6. And we thought that was kind of curious. And, Steve had a student that did a little bit of work. And, when we work with venoms in the lab, we want to kind of maximize the activity of some of the toxins so that we can kind of put a number on the activity levels that we get within a specific venom. And when you're designing this, you have to know, okay, what pH are you going to run this test at? And we found that the optimum pH, or like where the activity is the highest, is not at 5.5, but it's up at around 7.4 to 8. And so it kind of sparks this question of, okay, why are they acidifying the actual venom? Like, why is there acid in there? Why is it slightly acidic? And B, kind of, if there is a reason, is it a functional reason? Is it a vestigial reason? And how, kind of what does that really do for them? And, any test that you do on venom that is kind of straight out of the gland in a tube on its own, what we call crude venom, is you don't see any activity at all. Like the enzymes aren't functioning, the toxins aren't folding properly, and so they don't work. And that is really important because it means two things. Number one, the venom cannot really harm the animals while it's in the gland. And number two, some of the toxins that like to tear themselves apart when they are active are not doing so. So it is a mechanism that increases stability and increases safety. And we thought that was super cool.
Yeah. That was going to be my next question. You used the word safety earlier, and I was like, safety? But I guess that makes sense. If these venoms are breaking tissues down or doing whatever it is the particular venom does, like, how do they prevent it? So basically you learned, your master's thesis answered that question, why these venoms are slightly acidic, even though that's not the optimal number you would expect. And and you learned that it preserves the safety of the snake and helps essentially with the storage of the venom. Is that correct?
Yeah.
And then how does that, so if it's, these are probably very elementary questions for you guys. This is not your master's thesis defense by any means, but I know very little about venom. So if the snakes manage the pH to keep themselves safe, how then, when they inject it into a prey item, say, how are they not lowering the potential impact on that prey item? You know what I mean? Is it a whole... Yeah, explain that to me, because I'm. Yeah.
So this is probably like when I first like kind of put this whole thing together, I kind of got chills because it's one of those things that I just find so interesting. And if you kind of think back to, if anyone has ever taken human anatomy or human physiology, or really any mammalian physiology class, the pH at which our body sits at homeostasis, the vast majority of our tissues, whether it's muscle, blood, anything besides like the stomach or some of the filtration organs like kidneys or liver, they sit at a pH of around 7.4. And so what makes this so cool is that snakes have developed a system that keeps the venom dormant, and they don't turn it on. We do when it gets injected. Our body supplies the perfect environment for these toxins to kick on and just go crazy. And that's in my head, that's such an elegant solution to a fairly complicated problem.
Yeah, that is really cool. I hope people are following that, because it is very cool. And do we know if there's obviously lots of different types of venoms? I mean, every species obviously is unique. Sometimes there's variation within species, either spatially, throughout their lives that we discussed. Like, do we know if that is similar, let's just say, to different other rattlesnake species, maybe moving over to like elapids?
So as far as we understand, that kind of pH system is much, much more significant for enzymatic toxins. So these are toxins that are much larger. They're primarily found in vipers. So things that are causing tissue damage, things that are kind of kicking off reactions and tearing things apart. Those toxins really, really rely upon this. There are many other toxins. I think Steve talked about three-finger toxins and most neurotoxins that are not what we refer to as these enzymatic things. They are simply like a cap that goes on a neuron to stop it from receiving a signal or sending a signal, right? So they don't rely upon, their shape to tear things apart as much. They're a little bit more resilient to changes like pH. So this mechanism is most certainly viper and enzyme user affiliated. So definitely not as important for
lapids. So in most elapids, say, just generally saying neurotoxic snakes, it's less the pH isn't important in the same way. What they really need to do is just kind of keep that venom contained and outside of the system where it could attach to neurons and whatever nervous system components. Okay.
Yeah, exactly.
Gotcha. Well, that is really cool. I hope people follow that. So, great. So you're working on your master's, and you find out this really kind of cool result. And in the introduction, I already mentioned that you are currently a PhD student at Northern Colorado, and so you've done both your graduate degrees. I just wanted to, again, along the lines of advice, taking a little side here, and Did you have any thoughts? Maybe because you were so focused and there's just honestly so few people out there doing the types of things you're interested in. This wasn't even a factor, but like, had you put any thought to the concept of, some people do all of their degrees at one university or really spread them out? I mean, obviously you did your undergrad in Arizona. But did you put any thought to, before you continued on for a PhD, like, oh, I should go to another school, or again, was it just this is the lab to be in? I just need to stay at the same university just to show on my CV that I've been to 3 different universities is not worth, like, what was the whole thought process there, or was there a thought process?
There definitely was. The original idea was not to stay in the lab. The original idea was to go and work with other people. I think even just a year and a half as a master's student in Steve's lab, you get the opportunity to travel to other conferences and meet so many other people. And, unlike when I was at a conference when I was 16, going to conference as a master's student who actually has research, it's much easier to approach individuals and talk to them and like have, more, I guess, productive discussions as someone who's obviously active in the field. And, I had met a lot of people and I had plans to go somewhere else, and mostly because Steve had been pretty vocal that he was looking to retire. The last thing he wants to deal with is another PhD student, dragging him down. He's quite an interesting character when it comes to like trying to get a serious answer out of him sometimes can be a little difficult. But he kind of sprung it on me, the idea of sticking around for another 4 more years. And it hadn't been something I really considered until he mentioned that. And we talked a little bit about what it would look like and what projects we were interested in. And, how to really make that a productive, time, and what would we try to get accomplished. And so that it was not the original plan really until that conversation happened. And in fact, we were doing venom extractions. And he just kind of brought it up. He's like, what are you doing after this? And I was like, well, I'm going to probably, go to another lab. And he was like, oh, really? He's like, I thought you were going to stick around. I was like, last time we talked, you said you were going to retire, but whatever. So it's been phenomenal. And I definitely have no regrets at all.
Yeah. So you finish the master's and you go on to your PhD in the same lab. And this, I will say, this type of thing happens often. People just transition from a master's to a PhD. Sometimes it's kind of a continuation. I would say oftentimes it's kind of a continuation, like their master's research then kind of gets rolled up into something bigger for the PhD. But was that the case in yours, or did you kind of, obviously you're working in a Venom lab, so you're still working in the same general area, but was there a big shift in your research, or did you build on some of this pH related work that you did for your master's?
There was kind of a sizable shift. So the pH work was super interesting. I had such a, I will say I had a fun time doing it, despite like some of the hiccups that we had along the way. But it kind of put a cap on the end of, a long line of research into venom gland physiology and stuff like that. So it was kind of a good like little period for the time. And Steve and I actually got involved in a bunch of different other research projects my first year as a PhD student, and that kind of took a lot of attention. And they were all projects focused on, the treatment of snake bites, where we came from with treating snake bites and where we're going kind of treating snake bites, and some of the intricacies and complexities that we're, both facing and trying to innovate to kind of fight that challenge. And that's really where the research has gone, is it's moved away from, looking at snakes themselves, and it's gone much, much further into, we know what venom is. How do we start tackling the problem of envenomation?
Okay. So you start your PhD first year, you're kind of working on all these projects. And then, so first of all, how far into the PhD are you, just time-wise?
So this is my 4th semester in the PhD. Okay. So only about 2 years at this point.
And so, but you've probably developed your proposal for your dissertation research, and you're well underway. So how did you go from kind of this diversity of projects and, this shift of related to human and human health and snakebite and all of that? How did you develop the project, and what is kind of the the abstract to your dissertation proposal, essentially?
Yeah, so the start of this story really kind of goes back to before my undergrad, when I was still in Arizona, just a punk kid working with snakes at a reptile sanctuary. We had the Phoenix Herpetological Sanctuary has quite a few exotic venomous animals, and we actually did not have any exotic antivenom until about 2016, 2017, I believe, is really when we started the process of becoming an accredited antivenom bank. And I was very fortunate where they could just kind of let me sit in on the meetings. And I met two individuals that to this day are really, really big influences in my life. One is Dr. Michelle Ruha, who worked at the Banner Poison Center, the Banner Toxicology Center, was part of the Arizona Poison Center. And then Dr. Leslie Boyer, who is at the University of Arizona, is one of the most kind of notable figures in toxicology in the U.S. And I got to kind of talk with both of those people, and I really got a good idea of, what is antivenom? What does snakebite treatment look like? What are the issues that are facing it? And so I always had this kind of side interest in the medical stuff. And when I kind of started my PhD, kind of coming full circle, Dr. Leslie Boyer and a couple of other individuals, started collaborating with us. And, we got to kind of go into some of the nitty gritty on, new potentials for treatments and, what do those treatments look like and how do we fill in the gaps that we currently have in modern, envenomation treatments. And so it really came from an early passion that then kind of came full circle to reuniting with those individuals that have played a big part in my younger years. And so we're currently tackling a couple major problems. Number one is, yes, we're pretty good at treating snake bites to the point where we're good at saving lives, but we suck at saving morbidity. So mortality, death rates, incredibly low. Whenever you have antivenom, death rates are shockingly low. I think in the US, it's probably about 0.01%, something like that, fatality. And those are often extreme cases. However, tissue damage is severe and individuals often need to go through physical therapy. And there's much more nuanced issues that come with envenomation. So how we tackle that has really kind of taken the forefront of our attention.
Okay. And so how does your dissertation work address that? What's the plan for you to I'm assuming your dissertation work is focused around that morbidity component. And so what is the work you're doing and how is it going to inform our ability to lower morbidity rates?
Hey everybody, I just wanted to take a break and tell you guys that snakes are one of the most persecuted groups of animals in the world, but we are dedicated to changing that. The Orianne Society works every day to ensure there's a future for these amazing animals. If you care about snakes, wildlife, or wild places, and you like what you've been hearing on the Snake Talk podcast, support us today by donating on our website at www.orianne.org.
So I think the most important thing is we need better ways of assessing our treatments. If you look at, the FDA paperwork for Crofab and Anavip, the two antivenoms that we currently use in the United States, you'll find that the data that is used there is almost entirely survival rates. And so that's really all that assesses. If there's any data really available, a lot of times you have to find, case reports or case studies like that. And so what we're trying to do is how do you take something as complicated and nuanced as an envenomation, which obviously can differ. Like someone bitten on the hand is different than someone bitten on the foot, and what species and how much venom and what was, what population within that species and what's the age of the patient. So there's so many different layers to kind of quantify, and it can be really, really difficult to do so. And so we don't really have a model that is very good at kind of getting to those nuances. And I think, if we have a model that's accessible by, pharmaceutical companies or, by independent researchers where we can really kind of get at some of the details, that would be immensely helpful in assessing, what is the true quality of a treatment, and will it, fill the gap that we have. And so one of the big innovations that I think many of us are really hoping that this takes off are these orally available molecules. There's been a lot of data and kind of headlines on verasplitib. It's by a company called Ophorex, headed by Matt Lewin. And like that is a really cool candidate because it targets a huge, very conserved toxin family. Not only that, but it's orally available, right? So this idea of one day having a pill that you can take while you're hiking and you got bit by a snake and not waiting hours and hours and hours for antivenom, but having something on hand, like a snakebite kit that actually works is kind of the idea I have in my head, right? Not this like tourniquet and a razor blade that they sell at REI, right? I don't, that does more harm than good, as we know, but having something that's actually efficacious for people out there, I think would be amazing. And so the beginning really went on how do we assess, different treatments and, what are the potential treatments that could fill in the gaps and kind of shoe in those down or the shortcomings, I guess, of current treatments.
So your dissertation is focused on assessing the effectiveness of these new treatments, like the example you gave with the oral medication, or is it like helping to develop new treatments?
So my dissertation specifically is looking at the development of assessments and how we better assess. Like we've been lucky enough to partner with groups like verasplitib, as well as groups out of Denmark that are testing new age antivenoms, and so we can kind of use their really, really cool new drugs to kind of test our model. Yeah. And kind of fine tune it a little bit more along the way.
And so you're testing that in the lab, meaning, I don't know, you maybe envenomate some other type of animal and you're testing drugs on them, or are you kind of developing the template for how you assess, like the process, or both maybe?
We're definitely very hands-on. So we are, doing things like hemorrhage assays where hemorrhage assays are very, very popular, where you are giving an intradermal. So you're basically giving a blister of venom on a mouse and you're assessing the tissue damage of the skin, right? And can you limit that? But that also has issues because, how often is venom going to be trapped in the layers of skin and not, injected into a muscle? And then, so how do you really create something that's not this very 1D look at part of the problem.
Mm-hmm. Yeah, interesting. Well, I'm sure you can't really share it. Sounds like this is, there's patents and all these kinds of things involved, and you may not know yet. It's early on, but any like sneak peek? Do you think we are soon going to have new treatments for snakebite that are maybe more accessible? People can carry with them. People can take in wild settings. I mean, what are your initial thoughts there based on what you're seeing and what you know?
So, man, I'm kind of a realist, and despite me being a realist, there are some incredibly promising things that even very, very recently have come out. I know Steve on the episode he did with you was chatting about mini binder proteins. The group based out of Seattle was working on those, and those were incredible. In fact, that paper made it into Nature, and I think one of the authors of that paper won a Nobel Prize, which is just like kind of mind blowing. And so things like that are really coming along. And kind of speaking of Nature, we just were part of a paper published in Nature last month, where we looked at a truly innovative antivenom. So probably like the next big step in antivenomics since antivenom was really created. I mean, antivenom's been around for almost 200 years, and the process by which it is made has not really changed. You take venom, you put it into a large animal, you take out that animal's blood, you now have a treatment that you just purify down and inject into a human. This work was a little bit more precise, where instead of using the animals to create your final product, use your animals to create a template. You have viruses sort out the template, and then you take the things that you want and you express it in bacteria or yeast cells, not bacteria, and they produce it in a lab. And you can be very, very specific on how you treat with it, and it's modular, and it has all these different benefits. And so we're seeing really a next generation of antivenoms and treatments. And I think, despite me being a realist, and sometimes that's, kind of taken as pessimism, I think, we will be seeing some really, really innovative treatments here, within the next decade or so. And I think the treatment innovations will not only be in the hospital, but I think they will also be in the field where, you're really going to reduce snake and human conflict. And I think that's my big drive behind it. Is I want people to like these animals just as much as, you and I do. And, first step to getting there is, make the threat of a snake bite something that people don't even think about.
Yeah. So, last question there, and then I want to talk a little bit about the future. But so, as I'm sure you're well aware, there's a lot of inequality in snakebite treatment, and if we look at a global scale. So, if we look at different countries, we look at different socioeconomic classes, poverty, however you want to classify it. You could just take like the continent of Africa, for example, and while snakebite's kind of a big deal here, and we want better, innovative treatments, snakebite in places like Africa, parts of India, some other places, it's truly like a significant issue, a human health issue.
Yeah.
And so do you think these innovations, the way you see them coming down the line, are going to be just good for people like you and me sitting here in the US? Or do you think some of this innovative approach and this over-the-counter, if you will, approach will also help snakebite treatment in these places that are really underserved?
So, yeah, it's such a pressing topic when we get into the conversation of snakebite. Is, it might be good for us with all of our amazing infrastructure, but you pan over to another country, and we are very fortunate in that we work very closely with the Asclepius Snakebite Foundation, the group headed by Nick Brandehoff, Jason Foltz, Jordan Benjamin, and Ben German, all those guys, a bunch of MDs that are all herpers, are incredible toxicologists, and they do some amazing work out in Guinea is where they really started, and then now they're doing work in Sierra Leone and the Congo and all over the place, and they're really productive. And when they get to a country like Guinea, and they are able to do so much with basic education, basic equipment, and really, minimal supplies of antivenom, but at least something. And, I have the opportunities to kind of talk with those guys, on occasion. And we talk so much about how, yes, mortality is a difficult thing to tackle, but a trained MD with, 4 vials of antivenom can save somebody's life pretty easily. The problem is that person may still, never have use of that leg or that limb ever again. And when you work in an agrarian society, or a society so kind of centered on agriculture, that is debilitating. It's a lifelong change, and you all of a sudden, like, you have no livelihood. And so what's the frustrating side of my work is that, being in the US and working with new pharmaceuticals means that if you want to one day get your drugs overseas, whether, they're going to be in the hands of locals or the hands of physicians, you need to secure the financial side of everything so that you can do that. Because unfortunately, where the need is, the money is not.
Yeah.
Right. And so that's the challenge kind of facing these pharmaceutical companies is, yes, the goal is to one day make it widespread, but in the meantime, you have to make it really appealing to, the United States, secure all the funding that comes along with it, and then you can think about other things. And unfortunately, in the US, we just don't really have, like, snakebite is not a problem. We've got 8,000-ish on average envenomations a year. Of those 8,000, about 2 to 3 deaths. Like, it's a pretty low figure.
Yeah.
Over there, it's millions of bites. It's hundreds of thousands of deaths. It's millions of morbidity issues.
Yeah.
So really compounding problems in that region.
Well, I'll be hopeful because just, some of these innovations that you've hinted at, things like just an oral pill, even just something as simple as that, there, money and all that involved, I know, but it just seems like a simpler thing to get to people, say, in parts of Africa and all that. So I'll keep my fingers crossed there. So before we begin to wrap up, let's talk about the future. So you describe both your master's and your PhD, and they're both in the Venom world, if you will, but it's actually quite different. So what does the future hold? Are you interested in staying in academia? Are you interested in staying more in the human snakebite realm, or more kind of, going back to the basic, if you will, venom research? What are you thinking for the future?
I think academia, despite all of its flaws and frustrations, I love teaching, and I can see myself definitely finding a home in academia in the long term. In the short term, who knows? Maybe I do a postdoc a little bit more abstract, but I love the idea of working to reduce human-snake conflict, whether that is directly looking at medical studies, or whether that's, focusing on the biochemistry of how, a venom works, and kind of going into something that Steve had mentioned where you're looking at bioprospecting or looking at potential uses for venom. Because I think a lot of times Steve and I get thrown into this, like, oh my God, we're doing so much work for the betterment of humankind, when in reality, we're just two kids who, grew up loving snakes, and we want people to like them too. And so anything that we can do, or anything that we can kind of figure out through research to make these animals more interesting, less dangerous, and just kind of easier for people to live alongside is always of interest. So I've always had interest in, like, the actual venom itself. I think I'll never lose interest in that. I always love kind of working with these groups like ASF on mitigating the kind of the burden of snakebite in rural regions. And so, ideally, I would love to do all of it all at the same time forever. But, we'll see how realistic that turns out to be.
Excellent. Well, you have heard some episodes, so you probably know what's coming next. But I like to hear all of my guests tell me a good snake story. So Sam, what's your best snake story?
This one's actually kind of recent. It's from this past summer, and it starts about 2 years ago. I was starting a tattoo with an artist that's pretty well known within the reptile community. Her name is Samantha Mancino, and she's based out of Florida and travels around. And she does incredible pieces. I mean, absolutely just a phenomenal artist. And her and I talked for a long time on the idea. And during my first tattoo session with her, I'd flown to Miami. She was talking about how, she had just gotten back from Peru. She was with this other PhD student who, was thinking about doing some collaborations on art and, for the organization that she's running. And lo and behold, while I'm sitting there being tattooed, this PhD student calls the artist from Peru. And, just they're chatting about some scheduling stuff. And the artist goes, oh, I have another, PhD student who works with snakes, like, on my table right now. Do you want to talk to him? And the graduate student, her name was Ella Gardoir. She's from the Mark Margrave's lab. She is now a PhD candidate. And she was like, "Oh, what lab are you in?" I was like, "I'm Dr. Steve Mackessy's lab." And she's like, "I have been meaning to send you guys an email. I want to work on some stuff. I think this would be like a super interesting collaboration." And so I was like, "Awesome, let's do it." And we ended up actually flying down to Peru, to Iquitos, and then up into the Amazon to Ella's field site, and got to spend 10 days with her just in the Amazon herping, collecting venom samples, doing all types of stuff. And this has been, at this point, this has been a year and a half since we talked to each other. And it was such a random interaction. And, obviously if you're in, the Peruvian Amazon, you want to find a bushmaster, right? That is like the holy grail of a snake that you can find when you're down there, especially from someone who likes venom. And, I had kind of resigned myself to there's no way we're going to find one, right? I mean, like, I'm going to be a realist about this. The chances of seeing one, pretty low. And, we get to night number 10. We leave the next morning early. We had been rained on pretty hard. The trails were all flooded. And we were like, well, it's the last night. Let's go full effort and just, try to get to this one like area of habitat that we know is pretty good for lachesis, or should be theoretically. And, myself, Ella, and another student named Shiv kind of joins us, and we go through, and we are like up to our waist in a flooded trail. It's just like trying to slog through it. And I end up missing a turn on the trail, and we are lost. Like, we are off the trail. We are somewhere we had not been. And we were slowly coming to that realization when we crest the hill, and about 40 feet down the trail is a 7.5-foot Lachesis muta, big South American bushmaster, just sitting there curled up right on the trail. And like that moment, like I had been kind of daydreaming the whole time of like, oh, I'm going to see bushmaster and say something very cool and suave. And I see it, and I just start stuttering and mumbling like an idiot, because when you see an animal like that, it's just, how do you control yourself? And it was such an amazing moment. We brought her back. She was so sweet and calm. We got a venom extraction, collected some ectoparasites, scale clippings, took plenty of photos, and got to like show the locals, a snake that they are very afraid of. And, their name for that animal is shushupe. And so, a lot of them had never even touched a snake before. And so we had her tubed. And so for some of them, their first time ever touching a snake was this 7.5-foot bushmaster. And they all had smiles on their faces, and they're all so interested. And like that, those interactions mean so much to me. And I think, that story starting, a year and a half prior and leading up to that was just amazing. I will never forget that.
Yeah, what an adventure! I'm jealous. That sounds amazing. So, anyway, Sam, well, I'm glad you came on the podcast. Thank you for your time today. I've really enjoyed it.
Yeah, thank you so much for having me.
Yeah, well, and I just wanted to thank the audience and tell everybody to remember, snakes are animals too, and it's a privilege to see one in the wild.